Morning. This is Tim Cloughesy. I'm the Director of Neuro-Oncology at UCLA. And this is part of our annual brain tumor conference that we hold for patients and for caregivers. So thank you all for joining us. We hope this will be the last time that we'll be remote, get back to our in-person meetings. There's lots of reasons why we enjoy having the in-person meetings. One, getting to see you all face-to-face, allowing you to ask the kind of questions that you need to ask, but also getting you together with your peers to understand some of the things that your peers are going through and being able to share that. It's always been a wonderful experience, and we just want to be able to get back to that. So we're hoping to do that next year. First, these are just some of my disclosures before going forward. And I want to just cover first just some of the special things about UCLA. One of them is that we are a member of this SPORE group. It's a grant, and SPORE stands for Specialized Program of Research Excellence. It's provided by the NCI. These are very competitive grants. The funding is $12 million over 5 years. These are kind of the crown jewel of any cancer center in any of the universities. There are 6 brain spores that include UCSF, Duke, MD Anderson, Harvard, Northwestern, and ourselves. And just to give you a sense of how, and you should know that there are spores that exist for pretty much every tissue type of tumor, so breast cancer, prostate cancer, etc. But to give you a sense of how difficult these are to get, in California, again, which represents about 10% of the United States in terms of population, there's only 4 spores. And again, we are one of those, and between ourselves and UCSF, we cover 2 of those. Half of them are in brain. So there's a strong effort moving forward. And to give you a sense of what's involved in these studies, there's a infrastructure that has to be set up. There's different projects, there's different cores, and then there's kind of developmental and research program approaches trying to bring on the next generation of ideas moving forward. But these projects, there are 4 of them, they all have to be translational. They all have to have a clinical trial. They have to take some idea that has come out of the lab and then be applying it into the clinic. And so I think the nice thing when you look at this is you'll see there's 4 different projects from us, and there's 6 different groups. So essentially there's 24 different projects moving forward in brain cancer, trying to come up with better ways of treating cancer, doing experimental trials, trying to get answers. So, you know, the totality of this group alone should be able to push forward and get some good things moving forward. The 4 projects here at UCLA, there's one involving the immune system. This is led by Dr. Liao and Dr. Prince. It's a vaccine approach. This, many of our patients have been involved in this trial, and it's combining a vaccine with a checkpoint inhibitor, PD-1 antibody. There's one having to do with radiation and why tumors escape from having cell death in the setting of radiation. And it's really moving into a protective state that occurs with radiation. And there's a drug that keeps it from going into that protective state. And this is using a dopamine receptor antagonist. This is led by Dr. Nyemphu and Dr. Pajunk. And there's trials getting ready to start there. There's a trial on EGFR and targeting EGFR. And this is something that I'm involved with Dr. David Nathanson. And this is a target that's present in over 50% of glioblastoma. There's been a lot of EGFR inhibitors that have been out there. We have actually developed, synthetically created a molecule that gets into the brain, goes after these tumors and hits EGFR in a way that should allow us to have the kind of impact that we see in lung cancer, breast cancer with targeted therapy in the setting of glioblastoma. So we're excited about this. This is just getting going into patients. So their phase 1 study has been started. So we hope that we'll have good outcomes associated with that. There's a IDH inhibitor, and you're going to hear a lot about IDH because IDH really is such a critical mutation finding, and it represents the development and the primary reason why many of these tumors form, and I'd say about half of them, usually the lower grade tumors. But even though they're lower grade, they're still devastating. And we have a research project with Albert Lai and Dr. Harley Kornblum regarding that effort. So in addition to these projects, there's cores, as you might imagine, and this also helps, it just helps bring together our group to focus on research, to make sure that all the biospecimens that are available to do the research, creating models, all of that incredible neuroimaging that we have, and the availability to store that and evaluate it, and a really great biostatistics and bioinformatics group. And it's really the totality of all of this that I should say this has been built over years, and it culminates in getting something like this spore, but really it's only important if we change the disease in some way. So that's, again, 6 different centers, four projects each, 24 shots on goal. It's a pretty impressive group of folks throughout the country working on this. So we're really hopeful that we'll have a good effect there. So I'm going to move forward into the kind of didactic portion of this, where we just generally are going to talk about brain tumors and then bring up specific issues about different types of brain tumors. Covering first about the incidence, how do these tumors form, and then what are the subtypes and how do we catalog these subtypes? What are the unique features that create these subtypes? And within these subtypes, are there different features that are vulnerabilities that may be present that will allow us to come up with novel treatments and considerations as we move forward? So I'll get right into that with regard to the incidence and the causes. And brain tumors, there's about, almost 20,000 newly diagnosed brain cancer diagnoses each year in the United States. 50% of these are gliomas, and 50% of the gliomas are glioblastoma, which we'll get into, which is a particularly difficult to treat tumor. It's the second leading cause of cancer death in young adult males between 20 and 40. A particularly, as I said, difficult disease, one that requires lots of attention, lots of people working on to try to make a difference. This just gives you an idea about the incidence. And what we have here is how many patients, or how many people, I should say, per 100,000 persons per year will develop brain cancer of some sort. And then it's broken down by age that you could see across here. And I think the important thing is it's uncommon. We're down here at single digit per 100,000 people, pretty rare. But then you'll see this increase occurring as age increases. So the likelihood, for instance, of getting a glioma, the increase is almost to 20 per 100,000 patients from the age of 75 to 84. So you can imagine if you're having people living longer, higher numbers of people living longer, there's going to be more brain cancer diagnosed in an older population, and that the median, let's say for gliomas, then will shift from what used to be around 50, 55 as the median age for people getting a glioma to closer to 64, 65. So this is something that is going to be important for us as we move forward, as we all live longer, that this could be a significant part of the population that we have to address brain cancer with. As far as the causes, there's really very few known causes. Probably the one most important one is exposure to radiation in childhood, and in particular in the brain. This is something that has been seen where later on in life, after getting exposure to radiation to the brain, which usually is performed because one has a tumor, but one can then develop a secondary tumor 15, 20 years later. It's not a high percentage of patients where this happens, but it certainly, that is one of the clear known causes. As far as viral causes, we don't really have a clear understanding with. I think with cell phones, it's been really difficult to understand what the impact is of cell phones. I don't think it's been great, but there are some evaluations that believe that some of the schwannomas that are located on the nerve of the 8th cranial nerve that might in some way be related to cell phones. But we also have these genetically inherited syndromes. I think one of the questions that always comes up when somebody gets diagnosed with brain cancer is this something I could pass on to my children? And for 95% of people, the answer is going to be no. But with these rare syndromes, neurofibromatosis, where it would be obvious that there's a family history, Li-Fraumeni syndrome, also most of these, there's a clear family history, and you can identify that ahead of time and know that one could be at increased risk for brain tumors. The other finding is that about 19% of patients have a family history of cancer. But the reality is that's pretty much what is out there in the general population. So again, there's not a lot known about how to prevent these moving forward, or how to identify them early and identify populations that are most at risk, other than these genetically inherited syndromes. One of the issues, I think, with any cancer, especially a difficult to treat cancer like glioblastoma, for instance, that we're looking at here, is about early detection. And the earlier you can identify the tumor, the greater the likelihood that you will be able to control it. This is just an example where you see on the left-hand side here, this is a scan, a contrast scan, and a T2 scan. And this patient had a seizure, went into the emergency room, and had the scan, and the scan was read as normal. Well, in retrospect, we could see a little area here that looks slightly abnormal. But then 3 months later, this is what shows up. So the reality is, it's going to be almost impossible, at least with a disease like glioblastoma, as an example, to find these early. And once we find them, they're advanced. So you could see it only took 3 months for it to develop this rather advanced area of contrast enhancement, you know, with the ring enhancing around the edges, central necrosis, surrounding edema, mass effect. This is an advanced stage that occurs in 3 months. So no early detection. So then we have factors that in general, like why aren't we doing as well in brain cancer as we are with other tumors? And there are issues about the brain. There are issues about the type of cancer that occurs in the brain. Number one, these tumors seem to have intrinsic resistance to chemotherapy and other conventional therapies. And this might be because they came from the brain. These tumors come from, as we'll get into, oligodendroglial precursors, astrocytic tissues, oligodendroglial tissues. The brain is resistant to cell death. If the brain died easily, we would have a big problem. So there's this intrinsic resistance to therapy that exists. The brain is important. It's not like other diseases. It's not like a lung tumor where part of the lung could be removed, part of the liver could be removed, part of the colon could be removed. The brain is critical and it's susceptible to damage from therapy. There's also limited self-renewal. We can hit the bone marrow hard and the bone marrow can renew, but the brain does not renew. It's very limited in that way. It's also protected by a blood-brain barrier, and this is a vascular barrier that prevents things from going into the brain, and it only takes select things into the brain, nutrients, etc. And most of the drugs that are made do not cross this blood-brain barrier, so this becomes a concern and a limitation. And then, although I'd say, this next one about tumor heterogeneity, it's like, there's different areas where tumors look slightly, or the cells look slightly differently. Every cancer really deals with that. So it's not unique, but there are some that don't. Lymphomas can be very homogeneous and then could be very responsive to therapy, easily targeted because it's so homogeneous. And then the final thing is that the brain does not like inflammation, and it's going to do everything it can to limit inflammation. Well, part of that inflammation, though, is our immune system trying to fight the tumor. So if that's being limited, or if our therapeutic approaches are along the line of immune therapies, that could be a limitation as well. So to get at how these tumors form, this is something that we do not understand well, and can only get some inkling of understanding when we study this in animals. And what we know is there are stem cells that exist as our brain is forming, and our brain also has stem cells that exist to slowly replace the cells over time. And these stem cells undergo cell division. And with each cell division, you have to copy the DNA, and sometimes there's not complete fidelity with copying the DNA, and there could be a mistake that's made. And if that mistake is made in, let's say, some critical gene, there's the possibility that over time, as that cell goes on, and if it's a progenitor cell, goes on to, let's say, an oligo progenitor cell, those are the ones that end up going on to develop gliomas, whereas those that develop along a path of neurons, they are less likely to develop into some kind of tumor. So there's a unique cell type that is likely the origin for these tumors. So then they go on and they start to grow. They multiply, they might invade. And as they're multiplying and invading, it's the beginning of creating a unique ecosystem for the tumor to live in. That ecosystem involves having macrophages around it, having other supportive cells around it, new blood vessels that form. And all of this supports the tumor growing and creates this microenvironment that has drug resistance, that has invasion, that has immune suppression, that is feeding on itself to grow more and is developing new blood vessels. And that's when we see it on a scan. So that's when we see it here, is when it's getting to that point where it's large enough, where it has this unique environment that it's growing. And that's what we get to study. That's when we could take a tumor out, try and understand what it is, and go forward. We can't do it ahead of time. We can't do it here when we're not seeing any of that going on. We can only do it here when we see that going on. And so we take it, and we have done in our field and characterized this tumor in addition to many, many other types of brain tumors across gene expression, DNA copy number, specific mutations, characterizing proteins that are present, all of this to kind of better understand what's going on in the tumor. And what comes out of that is we get to see if there are certain patterns that are forming. And these patterns become important because they may identify specific areas of vulnerability or nodes that are being activated in a fairly continuous way. You know, one of the things that we learned, let's say in glioblastoma, is that there are 3 main nodes that are activated. They're not always activated the same way, but they're always activated, and almost every single one. So it's either going after some of the targets that are part of that activation and are maybe a critical component of that activation, or going after the hubs, the downstream hubs, in one way or the other. And all of this just gives us ideas about thinking about new therapeutic approaches. We also understand something that happens as the patients go through therapy. So we initiate a therapy, and that therapy that we initiate changes this tumor microenvironment and changes the tumor. It may adapt to it and may come up with ways of getting around that therapy, but it's that information that also helps us get a sense of how we should be thinking about, how we should be anticipating changes to therapy, what potentially could be added to prevent it changing and just simply killing the tumor rather than evading it. But one of the things that we see here is this is a group of patients, each of these little colored either circles or squares when they were newly diagnosed. And this is the change that occurs with regard to gene expression at recurrence. There are some tumors where there's lots of change that occurs, it goes in a particular direction, and other types of tumors where there's not a lot of change after therapy. The ones where there's a lot of change, at least to date, those are the ones that are usually pretty resistant to therapy. There's other things that happen as well. The cornerstone of the therapies that we use today, you're going to hear from the radiation oncologist, you're going to hear from the neuro-oncologist, that radiation and chemotherapy is the cornerstone of treatment for brain cancer. And it is. It's the best therapies that we have, but there can be consequences and changes that occur at the time of resistance because these DNA damaging agents, alkylators and radiation therapy, damage the DNA and hopefully lead to cell death. But when they don't lead to cell death, there's an opportunity for additional mutations. And this is just kind of a setting where we have an untreated patient then gets treated with a temozolomide, and we see all these new mutations showing up in a particular area. And that's a potential consequence of that therapy. Even though there is a benefit to that therapy, the end outcome of that therapy, sometimes after keeping the tumor controlled for some period of time, is that we develop these changes, and we have to be thoughtful about them and think about if there are ways that we could take advantage of these changes that occur and if they create a vulnerability for us to move forward. This is another example of a patient who had foundation medicine testing. This is next generation sequencing. We like to get this on all of our patients because they identify specific markers that may be unique for us to go after. But the important thing here is that the patient was treated, got radiation and chemotherapy, actually had 2 years without tumor growth with a glioblastoma, and then had a second surgery. Well, that second surgery found that there was a lot of new mutations that showed up, and some mutations that were present there before that have gone. So we like to, again, try to understand how the tumor changes with our therapies, and then how to anticipate that change in some way. So with all of this information, there's this opportunity to do different types of subtyping of tumors and say, I have all this gene information, I have this DNA copy number information, I have these mutations. Does that mean that my tumor is now just a EGFR mutated tumor? Well, the reality is there's two things that are really important for using these molecular features to help us characterize tumors. And that has to do with, does it provide a different understanding about prognosis? Meaning, does this population live longer than this population? If so, let's separate them out and have unique subtypes. Or is there something unique about a marker that will be predictive of a response to therapy. So I'm going to show you a couple examples of first how some of these features and molecular features help us understand prognosis, and then move on to show you how some of these molecular features can help us understand that the therapies that we're going to give are going to be beneficial because it is a predictive marker. I'm just going to start with this. Which is a finding that was made around 2008. And this was because there was an evaluation taking place on doing whole genome sequencing. And they identified in some gliomas the presence of this IDH mutation. Nobody really knew the importance with it. Isocitrate dehydrogenase usually as part of the Krebs cycle, it's taking glucose, breaking it down into smaller parts, creating energy. And in this particular part, the isocitrate dehydrogenase, which is this here, takes this substrate, isocitrate, turns it into alpha-ketoglutarate and moves forward. Well, when there's a mutation, there's a normal allele, so there's normal isocitrate dehydrogenase, and then there's the mutated one on the other allele. So that means that what happens in these cells is that isocitrate turns alpha-ketoglutarate, and then the mutated form of IDH takes alpha-ketoglutarate and turns it into 2-hydroxyglutarate. This happens only in the cell. It doesn't happen in the rest of the body. And it just turns out that 2-hydroxyglutarate accumulates in the cell. It cannot be metabolized. And because it accumulates in the cell, it creates changes in the cell. And the changes it creates include making changes in the chromatin, which is how the DNA and how the chromosomes are kind of rolled up. And it depends by changing that, it exposes different parts of the genome to be utilized. So it changes the expression of the DNA. And by changing the expression of the DNA, it makes it more of a stem-like cell or more of a progenitor cell. And that makes it more like a tumor cell because other mutations then come along and then it turns into a cancer. So this mutation in and of itself was identified. And then we went about trying to find out what are the tumors that this existed in. And we found a couple of different things. We found that these tumors are usually lower grade, that they are usually located in the frontal lobe. They usually are larger when they're diagnosed. Patients usually have seizures, but not necessarily symptoms associated with the diagnosis, so it takes longer to find it. There's other unique features that exist with this tumor that's different, let's say, than an IDH wild type tumor that occurs along the ventricle. So there was something very unique. And there was also something unique about the timing of when these would show up. They tended to occur between the ages of late teens into young adulthood. That was the unique time that this showed up. So what happens to be occurring around that time is myelination of the frontal lobes. And it may be that it's only in these areas where there's myelination of the frontal and the temporal lobes that are occurring that this is the time when this forms, at least for these kind of teen and adult forms of gliomas that are IDH mutant. One of the really fascinating things that occurred with this is by understanding this IDH mutation, it allowed us to have a better prognostic understanding of what to expect from patients. Over here, before 2016, we used a methodology where we basically looked under the microscope, found features, and tried to say that this tumor was grade 1, 2, or was grade 2, 3, 4, etc. and whether it had oligodendroglial features or astrocytic features. And you can see that these are survival curves. This one looks pretty consistent, but everything else in here is a mess. So that mess that we see here is because it is not an accurate representation of where, what type of tumor it is and what the prognosis it is. But when we incorporate IDH mutations, in addition to other features such as 1p19q, we now have what is clearly oligodendrogliomas that are IDH mutated, astrocytomas that are so again, the finding that of IDH changed from this very complex setting to this more well-organized and highly differential and more prognostic evaluation. So what we have here is the IDH mutant oligodendrogliomas, the lower grade IDH mutant astrocytomas, the lower grade IDH mutant grade 4 tumors, and then the lower grade IDH wild type tumors, and finally the lower grade IDH wild type grade 4 or glioblastoma. This provided a much more clear path forward and way of evaluating patients and giving patients a better understanding of prognosis than what we had previously. And then there was the new criteria that came out in 2016. There has since been other subtle changes for the 2021, and Dr. Fausto Rodriguez will be able to go through that with you all. So that was the prognostic part. The predictive part was when you begin to divide these subgroups out. So in this case, these are all grade 3 tumors. These are the grade 3 oligos that are IDH mutant. These are the grade 3 astrocytomas that are IDH mutant. And these are the grade 3 astrocytomas that have no IDH mutation. Well, the two lines that you see separate here was a study where in these patients, some got radiation therapy alone, others got radiation plus chemotherapy. And so what this shows is in the IDH mutant tumor types, this separation occurs because of the addition of chemotherapy. This is showing the predictive value of adding chemotherapy to radiation therapy in patients who have an IDH mutation, whereas those who do not have an IDH mutation, there's very little impact of adding chemotherapy to radiation therapy. This was seen not only in grade 3 tumors, but also in grade 2 tumors, where if you separated out the IDH mutant group and you evaluated them, you would see that those that received radiation therapy alone did not do as well as those that received radiation therapy and chemotherapy. So again, this is a predictive test. So we saw the prognostic aspects of having molecular characterization. This is the predictive aspects of having molecular information. There's an additional setting where this takes place, and that's in the IDH wild type group. Well, this is the original study that showed that there was a difference between radiation therapy alone and IDH wild type glioblastoma and radiation therapy and chemotherapy. This improvement, although minor at the median, as the tail went forward, the amount of improvement increased. And this is the standard of care for patients who are undergoing treatment for glioblastoma. But when we look at MGMT methylation status, and this is just another biomarker, and MGMT methylation status just really says that the gene that makes MGMT, it has a promoter, and this promoter has all these methyl groups on it. And if it has the methyl groups on it, it's not being expressed. And if it's not being expressed, then the gene isn't being made into a protein, and that protein's job is when we give temozolomide and we cause an abnormality on the DNA, its job is to remove it. So MGMT methylated means that when we give chemotherapy like temozolomide, that there should be a good response, that the lesion that's made in the DNA stays, it doesn't go away. When it's unmethylated, that means that the gene is being expressed, it is turned into a protein, that protein goes and removes those abnormalities on the DNA caused by chemotherapy. So when we look at this gold and this kind of a more pale blue group, these two are MGMT methylated. This is the group that got radiation alone. This is the group that got radiation plus chemotherapy. So that's the benefit that we see of adding chemotherapy is to those that are MGMT methylated. That was not true in the setting of the unmethylated setting. What we see here is the red is those that receive radiation therapy alone. The navy blue is the group that received radiation therapy and chemotherapy. Both of these are unmethylated. There is a difference. There is a benefit to getting chemotherapy, but it's a minor benefit. So again, just to show this in a different way, this is the way that we can split out some of the prognostic and predictive markers associated with gliomas. First question is, are they IDH mutated? Next question is there a 1p19q codeletion? If there is, it's an oligodendroglioma, and it's either grade 2 or grade 3 based upon how it looks under the microscope. If it's IDH mutated and there's no 1p19q codeleted, then it's either grade 2, 3, or 4, and it depends how it looks under the microscope to get that grade. For those that are non-IDH mutated, the majority of those are going to be glioblastoma. They're going to be called grade 4 glioblastoma. There's some over here that we'll talk about in a second. But roughly 40% of these are going to be MGMT methylated, and about 60% are going to be unmethylated. They don't change their grade. Their grade is the same, but they may or may not be MGMT methylated. And that's the difference between, as we showed, the prognostic difference and the predictive difference. There's still others, and I'll let Dr. Fausto Rodriguez describe to you the issues around those that are histone mutated versus PXAs and others that are unique, where there may be therapies that can be effective in these settings. So by going about and separating these groups out into IDH mutated and non-mutated, there's other opportunities for treatments. And so if we look at the other features that we see associated with IDH mutant, or I should say non-IDH mutant tumors, we see this group along here. The majority of them are going to have EGFR. There'll be a number that just have other mutations such as NF1. And then there'll be this group that has a series of rare mutations. Well, in particular, this group here, these mutations have been found that in other tumor types, if they exist, that therapies can be used to treat this, to try to control the tumor, and they have controlled the tumor. So if we see this in a glioma, there's an opportunity for us to consider those other therapies if they could get into the brain well, and to move forward with these treatments. So that's one of the reasons that we want further molecular detail on every patient, for us to do MGMT, or excuse me, for us to do next generation sequencing on those patients. So in addition to the idea that IDH mutant tumors are responsive to radiation and chemotherapy, they also provide an opportunity to understand what's going on with these mutations and can they be specifically treated outside of radiation and chemotherapy. And that's when we get into this idea of the IDH inhibitors. So this is an example of a patient who has an IDH mutant glioma. This is a scan, and then a year or so later, we see that it increases in size, and then this patient initiates treatment with an IDH inhibitor. So the IDH inhibitor is given, and we see that the size of this is smaller, that the pressure that's placed on the ventricle is less, that the size of the cavity has increased because there's less bulk and tumor. So it's not only that existing drugs that are out there to try to match with mutations, this was actually created specifically because of this mutation. So this opportunity to treat gliomas differently occurred because it was recognized by doing next generation sequencing that IDH mutations occurred. Biology was done, and then it was found that we can inhibit these, and in these patients, we can actually get tumor shrinkage. So again, we went from prognosis to predictive therapies to even creating new therapies with this. So the same thing can be true not only for the IDH mutant tumors, but these other mutations. And to date, for H3K27M mutant tumors, there are these dopamine receptor antagonists. For BRAF mutant tumors, there's BRAF inhibitors and MEK inhibitors. For NTrack fusions, there's NTrack inhibitors. For FGFR, there's FGFR receptor inhibitors, etc. I want to show an example of how, and I think, just such an illustrative example of how identifying mutations that occur in other tumor types can be applied in the setting of gliomas. And this is with what are these track fusions or NTrack fusions. These are really rare. There's only about 5,000 total patients in the world or in the United States that have these, but every year that's what's happening. They identified through next generation sequencing that these fusions existed. They could exist in many different tumor types, and they exist in kids and adults. And this was the kind of effect that they would see when they would give an NTrack inhibitor in patients who had these fusions. So this is an example of the maximal change from baseline of a tumor. So this would be around a 50% increase. This would be 100% decrease. The majority of the patients treated with these have a decrease in the size of the tumor, some of them close to 100%. If we begin to break down the different subgroups, we see that there's different NTrack genes that can have fusion partners, and across the board, they're getting effects associated with that. We could also look at the different areas in the body where these tumors are occurring, and we have responses across the board. To give you a sense of how significant this is, I'm sorry, this seems a little graphic. This is a tumor that was exophytic coming outside of the body. The patient started treatment on an NTrack inhibitor. By 6 days, it had visibly decreased. By 21 days, it was almost gone. That's impressive. That's the kind of thing that we want to see. That's what we want to try to continue to develop for patients with brain tumors, but with all kinds of tumors. This is why getting next generation sequencing is so important for considering therapies in our patients. So we covered radiation therapy, chemotherapy to some degree. We talked about these targeted therapies. One of the hot areas is still immunotherapy in gliomas. And I think this is a really exciting area. I mean, just the idea that your body can control your cancer and maybe needs a little bit of help in doing that is just really fascinating. But we want to get into why there's benefit in some tumor types and why we struggle so much in the setting of glioma of getting immunotherapies to be effective. There's this something called the the immune therapy cycle or the immune cycle. And this is what could be happening in our bodies if there are no components of this immune system that is being inhibited or stopped. So we wouldn't even know that we were having a tumor controlled. It would just be controlled. And that is that there would be a small area of a tumor. It would release different proteins associated with that tumor. It would be picked up by something called dendritic cells or antigen-presenting cells. These would go to a lymph node, and then in the lymph node, these would develop unique T cells that would go against the targets on the tumor, the specific targets. Those T cells would then go throughout the body. They would surveil, they would go to the area of the tumor. They would have these specific targets link and interact and kill the tumor. And if everything's working great, if there are no deficits in this immune cycle, there's no tumor that we know about. But there's several things that activate and inhibit our immune system that is finely tuned, and we want it to be inhibited in some setting. We don't want our immune system going crazy and causing autoimmune disease, other things like that. So we want to be able to understand what triggers and what inhibits the immune system. There's a number of different things that exist out there. And when it's finely tuned, it works great. Not too much, not attacking normal organs, and not too little, getting rid of cancer. The most remarkable thing that happened, and it was really in this area down here where we talk about something called PD-L1 and PD-1, and that is when there's inflammation that occurs in a tumor. So we start this process and we go through it and we get inflammation in the tumor, and the surrounding cells of the tumor, there's inflammation. That is where we begin to have problems where the tumor and the surrounding microenvironment start to express PD-L1. And when PD-L1 is expressed, the T cells that are on there have a receptor called PD-1. And those T cells then go to sleep. When those T cells go to sleep, they're no longer killing the tumor, and the tumor can continue to grow. So although this is a way that has been set up in our body to prevent autoimmune disease, it also can allow for tumors to grow. And by simply using an antibody that blocks PD-L1 or blocks PD-1 or PD-L1, the T cells wake up, they expand, they start killing the tumor, and we have a benefit. That's what's seen in lung cancer, that's what's seen in melanoma. They have one deficit, only one immune deficit. That's at the area of the tumor where that immune deficit exists. In glioma, we have multiple areas of deficit. Our targets are not very immunogenic, meaning the tumors don't look that abnormal. We have limited ability to get antigen-presenting cells in the brain. The systemic immune system is suppressed just from the tumor itself, not in addition to the radiation and chemotherapy. We have limited infiltration into the tumor, and this tumor microenvironment just prevents the development of a good immunologic response. All of these deficits exist, and all of them need to not exist in order for us to have an active therapy. And or to have an effective therapy. And we have been evaluating vaccines, trying to go after better antigen presentation, viruses to get better kill and to get more immunogenic effects, CAR T cells, BiTEs, immune checkpoint inhibitors to change this microenvironment. All of them are being evaluated, but part of the problem is if you just use one of them, that's not going to be adequate because other deficits exist in these other areas. So just to be clear what needs to happen, if we're going to give a vaccine, the first thing that needs to happen is we have immunogenic peptides that we think are associated with the tumor. The tumor or the antigen-presenting cells take the pieces from that vaccine, show it to the lymph nodes. Then there has to be the development of the vaccine-specific T cells. Those T cells then have to infiltrate into the tumor and to begin to kill any tumor cells, hopefully, that have those specific epitopes that the vaccine is providing. We then, with that cell kill, more antigens are released, and there's more antigens that can be presented on these antigen-presenting cells. That leads to the more antigen-specific T cells that come from the cancer. And that leads to infiltration of the cancer antigen-specific T cells. And then hopefully, that's when we get cell kill. Again, all of those things have to happen. So you can get an idea that if there's deficits along the way, is probably going to require combination approaches. I'm going to end this with just some innovations that are occurring in the clinical trial space having to do with clinical trial design. And this is going to be gone into greater detail by Dr. Niemfu. But this is the platform trial, GBM Agile. It's called an adaptive platform trial. And what it is it evaluates patients with glioblastoma, either newly diagnosed or recurrent. And there are different therapies, and to date, there are currently 5 therapies that are in the trial. And they're all compared against the same control over time. So instead of running separate trials where everybody has a control, and most of these trials, it's a 1-to-1 randomization. That means 50% of the patients get experimental therapy and 50% get some kind of control or standard therapy. When you have multiple arms in one control, the control could get 20% of the patients, and the rest of these get 80%. That's a much better randomization that favors the benefit of patients getting this. The second thing that occurs is within these evaluations, there's competition. So for the experimental therapies, as the study is ongoing, it has an algorithm. And the algorithm finds out for a specific patient going in, as they're getting ready to be randomized, it gets compared, and that specific patient, let's say they're recurrent. How's the group from regorafenib doing who's recurrent? Or for Paxlovid, or Val-083, or VT1021, or Troriluzole. And the one that's performing the best gets the greatest percentage of that 80%. Doesn't get all of it, but it gets the greatest percentage. That means any patient coming in has those unique features are more likely to get the therapy that's best for them. And this moves forward, and any drug that's successful in this setting moves forward and can have a new drug application, which means it could be marketed, FDA approved, and not only in the US, but this is being done in Canada, Europe, going to be moving into China and Australia. All of this provides a great opportunity for faster, cheaper, fewer patients. We want these evaluations to occur quickly. We want shots on goal. And this is a mixture of lowering the cost so that more companies want to come in to get an opportunity to have success in glioblastoma. So I'm going to stop here. And again, I just want to thank you all for joining us. I hope you enjoy the rest of the conference. And again, we look forward to seeing you in person soon. Take care.